Hello students, in this PowerPoint presentation I would like to discuss the second experiment of B Pharmacy 6th semester, that is anti-allergic activity by mast cell stabilization assay, right? So here we will discuss all the experiment in detail. The requirement for this experiment, we require the guinea pigs, albino rats, and the drugs like histamine chloride, chlorpheniramine maleate, and the doses are specific. So students must have to learn what are the doses that they are using for this experiment. Like reagent, we will use RPMI buffer, egg albumin, toluidine blue solution we will use the instrument like microscope with 10x magnification. So in the further discussion I will explain what is the role of each drug, why we are using egg albumin, toluidine, and why we are using saline solution in detail. So this is the requirement for this experiment. In the second slide we will see about theory. As we are checking the mast cell stabilization, we have idea about the mast cell that it has the Fc silane R1 receptor as it is represented in the diagram, and as soon as any kind of allergen binds with this Fc silane RA receptor on the mast cell, they become in the dimeric form, means two Ig antibody combine cross links, and further there will be the activation of transducer mechanism that leads to the degranulation of histamine granules. So the mast cell, once stimulated by the allergen, responsible for mainly release of histamine, but along with the histamine like tryptase, PAF, and the proteases, cytokines, chemokines, nitric oxide, and then endothelin are also there. But basically we will talk about the histamine. As we know, the histamine the histamines is responsible for exposing the bronchoconstriction. If we talk about the bronchioles, the histamine is a histamine considered as secondary mediator, is autocoid and responsible for bronchoconstriction. So this is the basic idea that if we expose any animal to the histamine, the bronchioles of the animal will become contracted, and when the bronchioles of the animal become contracted, the animal will not respire freely. Animal will feel like shortness of breath and dyspnea, right? So in the present study is designed to explain that histamine causes bronchoconstriction and therefore responsible for pre-convulsion dyspnea, in short we say PCD. So we will use in this experiment like various drug that actually stabilize the mast cell. The meaning of stabilization of mast cell is that once the mast cell becomes stabilized, now the mast cell will suppresses the release of histamine. So once we using like disodium chromoglycate drug is here, which is used as the mast cell stabilizer. So when we use disodium chromoglycate, there will be suppression of histamine release from the mast cell. Here I represented the diagram which is of histamine chamber. So student can see there is a meter, right? This is the this one is the meter where you can where we can see the ultimate pressure, and from here we will generate the pressure. Here is the how much volume in which we are exposing the animal, and this is the glass like structure which contains the histamines. So we in a controlled way 0.2% histamine will expose to the animal. Animal ko hum will put animal in this histamine chamber and we will expose animal to the histamine 0.2%. Later on we will discuss in this point in detail. So first, in the mast cell stabilization assay, the first mast cell stabilization, we will do the mast cell stabilization experiment by evaluating the bronchoconstriction through the histamine in the guinea pig, right? बाय यूजिंग हिस्टामिन एरोसॉल। तो यहाँ पे हम हिस्टामिन एरोसॉल का यूज करेंगे और कॉन्स्ट्रिक्शन का इफेक्ट देखेंगे इन द गिनी पिग। तो फर्स्ट क्वेश्चन इज दैट व्हाई वी आर यूजिंग द गिनी पिग ओनली, नॉट वी आर टॉकिंग अबाउट द रैट इन दिस एक्सपेरिमेंट और माइस, because the selection of animal is based on the sensitivity to that particular drug. If we see the sensitivity of histamine to the guinea pig bronchioles or the respiratory system is higher than other animal, that's why we are using the guinea pig here, not any other reason. So in this experiment, well, first we will take the guinea pig, that will be number of 6 guinea pig will be there. We will divide these 6 guinea pig into 2 group, and each group contain 3 guinea pig, right? All group means group 1 and group 2. Group 1 and group 2, all the guinea pig will be exposed to the histamine chamber, like you can see here. This is the, we are exposing the histamine chamber here. Histamine where will be 0.2%, right? And here we will check the time for pre-convulsion dyspnea. मीन्स हियर वी वंस, यहाँ पे हम बेसिकली देखते हैं प्री कन्वर्जन डेस्पनिया का टाइम। तो प्री कन्वर्जन डेस्पनिया होता क्या है, लाइक हमने हिस्टामिन 0.2% जिस टाइम पे एनिमल में एक्सपोज किया, और एनिमल में उस हिस्टामिन की वजह से जब ऑनसेट ऑफ कन्वर्जन के पहले का जो टाइम है, मीन्स उसमें जो जर्किंग होगी, स्नीजिंग होगी उस एनिमल में, उसके एब्डोमिनल का जो मूवमेंट वहाँ पे जर्किंग दिखेगा, वो कन्वर्जन के जस्ट पहले की स्टेज। मीन्स हिस्टामिन एरोसॉल 0.2% को एक्सपोज करने से लेके जब तक कि एनिमल को कन्वर्जन आने वाला हो, मीन्स जस्ट ही ऑनसेट ऑफ कन्वर्जन जब स्टार्ट हो, उसके बीच की स्टेट, मतलब ऑनसेट ऑफ कन्वर्जन के पहले और हिस्टामिन को एक्सपोज करने के बाद की जो बीच का गैप है, उसको that is called pre-convulsion dyspnea. So we will determine the pre-convulsion dyspnea. Now once we will determine the pre-convulsion dyspnea, we will remove the animal from the histamine chamber and we will put the animal in the fresh air, and after 24 hours we will take the group 2, right? Here in the group 2, we will administer a drug which called chlorpheniramine 2 mg per kg subcutaneously. So it is important to know that why we are administrating chlorpheniramine, because chlorpheniramine is antihistaminic drug and it does not allow the histamines to bind histamine to perform their action, right? As we know, the pre-convulsion dyspnea is just because of the histamine, and when we will use the chlorpheniramine in the group 2 only, not we are in the group 1, we will not use chlorpheniramine. So once we will in group 2, when we use chlorpheniramine, after 1 hour of drug administration, again we will put one by one guinea pig of group 2 into the histamine chamber and again exposed to the 0.2% histamine aerosol. Now we will determine again the pre-convulsion dyspnea. So student can understand from here कि जब हमने हिस्टामिन दिया एक ग्रुप को और उसको हिस्टामिन चैम्बर से एक्सपोज कराया, तो उसका एक पर्टिकुलर पीसीडी हमने नोट किया, और ग्रुप 1 को जब हमने कुछ 24 आवर्स के बाद फिर से हमने उसको हिस्टामिन चैम्बर में रखा, तो बट ऑब्वियस सी बात है उनका जो पीसीडी वैल्यू होगा वो सेम होगा। लेकिन ग्रुप 2 के केस में जब तक हमने उसको हिस्टामिन चैम्बर में रखा, जब तक हमने ड्रग नहीं दी थी, उसके पहले की पीसीडी वैल्यू और ड्रग देने के बाद वाली पीसीडी वैल्यू में डिफरेंस आ चुका होगा। पीसीडी वैल्यू बढ़ चुकी होगी इन द सेंस, अब उसको अगर सबसे पहले हम बात करें ग्रुप 2 के किसी एक एनिमल की, तो अगर पीसीडी वैल्यू पहले 5 मिनट, लेट सपोज फॉर एग्जांपल अगर हम बात करते हैं 5 मिनट्स है, तो वो अब मोर एंड लेस वो हो, लेट सपोज कि वो 6 मिनट हो चुकी होगी, 5 मिनट से 6 मिनट, तो 1 मिनट का ये जो टाइम स्पैन बढ़ गया पीसीडी वैल्यू आने में, प्री कन्वर्जन डेस्पनिया को आने में। तो वो उसकी वजह ये थी कि उसमें आपने एंटी हिस्टामिनिक्स ड्रग का एक्शन था, जिसकी वजह से हिस्टामिन ने अपना एक्शन प्रोमिनेंट जल्दी से नहीं दिया और वो उस 5 मिनट में हिस्टामिन का इफेक्ट नहीं आया। क्लोरफेनिरामिन देने से हिस्टामिन का इफेक्ट थोड़ा प्रोलॉन्ग हो गया, तो हमने कहा कि पीसीडी वैल्यू इसकी बढ़ गई। और एट लास्ट वी विल डिटरमाइन द परसेंटेज प्रोटेक्शन। सो हाउ वी विल डिटरमाइन द परसेंटेज प्रोटेक्शन? हियर इज द formula explaining that 1 minus T1 by T2 in the bracket, inside bracket, will multiplied by 100. What is the T1 here? T1 is the mean value of PCD. Why we are seeing the mean value? Because we are taking the guinea pig, not single guinea pig. We are taking guinea pig more than one. T1 is the before drug administration PCD value and T2 is the after drug administration PCD value. So once we will calculate the percentage protection, we will draw observation table in which the observation table, the group and the percentage protection. Group 1, which we were administrating only the saline, not we were exposing these animal to the chlorpheniramine. So here the percentage protection, probably it might be 0, and in the group 2 chlorpheniramine maleate according to the data, framed data. So observation table will be like this. And in the second way, so this is the first one, evaluation of bronchoconstriction in the guinea pig using histamine aerosol. Now in this second point, to mast cell stabilization assay. In this assay, we will take 6 albino rats and divide into 2 groups. Each group contain 3 rats. So up to the 3 days, 3 दिनों तक लगातार group 1 will receive the normal saline while the group 2 animal receive disodium chromoglycate 50 mg per kg intraperitoneal. So 3 दिन तक हमको ये दोनों ड्रग्स देनी है. ग्रुप 1 में सलाइन देना है और ग्रुप 2 में डाइसोडियम क्रोमोग्लाइकेट विच इज मास्ट सेल स्टेबलाइजर. तो मास्ट सेल स्टेबलाइजर है, इट मीन्स ये हिस्टामिन को रिलीज नहीं होने देगा. राइट, हिस्टामिन को रिलीज नहीं होने देगा, इट मीन्स डीग्रेनुलेशन नहीं होगा. ग्रेनुलेशन के फॉर्म में ही जो है वेसाइकिल रहेगी मास्ट सेल के अंदर हिस्टामिन के जो पार्टिकल होंगे. तो ऑन 4th डे, 3 दिन तक हमने ये प्रोसीजर किया. ऑन 4th डे वी विल इंजेक्ट 10 ml per kg नॉर्मल सलाइन टू ईच एनिमल ऑफ ईच ग्रुप. दोनों ग्रुप में जो 3 एनिमल थे, मीन्स 6 रैट को हमने चौथे दिन 10 ml per kg के हिसाब से नॉर्मल सलाइन एडमिनिस्टर करना है. नाउ विल वंस आफ्टर एडमिनिस्ट्रेशन वी विल मसाज टू द पेरिटोनियल रीजन फॉर 5 minutes and after that we will collect the peritoneal fluid in the test tube from the abdomen, and once we will collect the peritoneal fluid, we will transfer this peritoneal fluid in the test tube already containing RPMI buffer 7 to 10 ml. 7 से 10 ml ऑलरेडी एक टेस्ट ट्यूब में RPMI बफर होगा, जिसमें हम लोगों को जो पेरिटोनियल फ्लूइड, एनिमल के पेरिटोनियल फ्लूइड से जो हमने कलेक्ट किया उसको उस RPMI बफर वाली टेस्ट ट्यूब में ट्रांसफर कर देना है. नाउ वी विल सेंट्रीफ्यूग दैट ट्यूब एट ए स्पीड 400 RPM. राइट, नाउ after once centrifugation is done, now the supernatant will be discarded and we will if we want to wash the twice that remaining things, we will add the RPMI buffer once again. And the purpose of washing is that actually we are washing the pellets of mast cell. तो अगर मास्ट सेल को वॉश करना है तो RPMI बफर डाल के फिर से उसका सेंट्रीफ्यूगेशन कर से और फिर से उसको सुपरनेटेंट को डिस्कार्ड कर देंगे. Now there is an important step that we are adding in this step, egg albumin in the cell suspension, and we will incubate this assembly after adding egg albumin to that particular tube. We will keep that things in a 37 degrees Celsius for 10 minutes. What is the purpose of egg albumin? Why we are adding here? Egg albumin is considered as the allergen in this activity. So the mast cell which is in the test tube in the peritoneal fluid is once we will add the egg albumin, that particular mast cell present in the test tube considered this egg albumin as an allergen, and once it considered as the allergen, it will start degranulating, releasing histamine. Right, so the purpose of adding egg albumin to exposure of allergen. Right, now here we will stain with 1% toluidine blue solution. Why we are taking toluidine? Because toluidine is basic in nature and it attaches to the acidic compound and acidic compound of the mast cell like histamine. So now we will observe the slide under the microscope. We will calculate or count the number of granulated and degranulated mast cell in a different visual area. At least we have to count the 100 mast cell in a visual area. In the next point, जब हम इस 100 mast cell को calculate कर लेंगे So in the we will draw a table that containing group 1 normal control में whatever the granulated amount will count and whatever the degranulated we will write it here, and after the disodium chromoglycate. So the disodium chromoglycate को ऐड करने के बाद, चूंकि ये मास्ट सेल स्टेबलाइजर है, अगर मास्ट सेल को जो चीज स्टेबलाइज कर रही है, इट मींस वो डीग्रेनुलेशन को प्रमोट नहीं कर रही, मींस यहाँ पे डीग्रेनुलेटेड कम होगा और ग्रेनुलेटेड ज्यादा होगा इन द ग्रुप सेकेंड. So from this conclusion, the conclusion, we have reached to the final that the pre-treatment of animal with mast cell stabilizer stabilizes the mast cell by rendering it in hyperpolarizing state that inhibit release of secondary mediator like histamine, which is responsible for vasoconstriction. This will be our conclusion. And how we will write the result of this experiment? And the result of this experiment we will write in such a way, like in evaluation of bronchoconstriction in guinea pigs using histamine aerosol, percentage production for group 1 is this and for group 2 is this. This shows the antihistaminic action of chlorpheniramine. And for in mast cell stabilization assay before exposure of disodium chromoglycate, the granulated and degranulated particle number is this, and after exposure of disodium chromoglycate, the number of granulated and degranulated particle is this. So this will be the result. And at last, after a student study about this mast cell stabilization, each student must know that each student must able to define what is pre-convulsion dyspnea, that I already discussed during the presentation. This pre-convulsion dyspnea, what is the mechanism of sodium chromoglycate, and what is the use of toluidine, why we are using toluidine in the mast cell stabilization, and what is the role of egg albumin. Student must add these questions in the practical records. So this is the references where I from where I taken this content, and this is the this one is the, I hope कि आपको ये experiment पूरा clear होगा. So this is all about the anti-allergic activity by mast cell stabilization assay. Thank you very much.